Welcome to the lecture on kidney autoregulation. My name is Olga Sosnovtseva, Department of Biomedical Sciences, University of Copenhagen. Adults have around 7-8 liters of blood, and this is constantly filtered by the kidneys, as much as 400 times per day. The kidneys reabsorb and distribute 99% of the blood volume, and only 1% of the filtered blood becomes urine. Nearly 500 million people globally, which is about 10% of global adult population, suffer from some kind of kidney problems and damage. The kidneys serve multiple functions, including regulation of water and electrolyte balance, excretion of metabolic products, secretion of hormones, and regulation of arterial pressure. Interaction between the kidneys and cardiovascular system is complex. On one hand, the kidneys provide important long-term regulation of the cardiovascular system by maintaining the volume and composition of the body fluids within narrow bounds and by producing a set of hormones that affect blood vessels within the kidney and elsewhere in the body. Disruption of kidney function can cause, for example, hypertension. On the other hand, the kidneys are also perfused with blood and therefore exposed to all of the fluctuations present in the cardiovascular system. To maintain its stable functioning, the kidneys possess some regulatory mechanisms. Functional unit of the kidney is called nephron. This figure illustrates the main structure of the nephron. Blood enters the system through the efferent areola, a short vessel that is capable of regulating the blood flow by varying its diameter. At the glomerulus, the blood passes a system of parallel capillary loops where 25-35% of water together with blood elements with low molecular weight is filtered out into the tubula. Blood cells and proteins are retained. The filtration process saturates when the osmotic pressure balances the hydrostatic pressure difference between the blood and the filtrate in the tubula. The blood leaves the glomerulus through the efferent areola to pass through a new capillary band and out to the venous system. The other component of the nephron is a hollow tube, marked in yellow in this figure. There are different sections: the proximal tubula, the loop of Henle, the distal tubula, and the collecting duct. Here, processes of reabsorption and secretion take place. The proximal tubula is located within the outer layer of the kidney, the cortex, and therefore it's accessible for different measurements. Except that there are no large molecules, the composition of the ultrafiltrate in the proximal tubula is similar to the blood plasma. Without changing the composition much, the proximal tubula reabsorbs approximately 2/3 of the water and salts. From the proximal section, the tubula passes down through the renal medulla, forming the descending and ascending limb of the loop of Henle. As the filtrate flows into the descending limb of the loop of Henle, the concentration of sodium chloride in the interstitial fluid surrounding the tubula increases significantly, and osmotic processes cause a reabsorption of water. At the same time, salts and metabolic products are secreted into the tubular fluid. The ascending limb of the loop of Henle, on the other hand, is practically impermeable to water, and its cells contain molecular pumps that transport sodium and chloride from the tubular fluid outside. In this way, the sodium chloride concentration in tubular fluid is reduced. When re-entering the cortex, the ascending limb becomes the distal tubula. It is the prime site for the kidney hormone-based regulation of calcium. In collecting duct, further reabsorption and secretion takes place. The interesting anatomical feature is that the terminal part of the ascending limb of loop of Henle and early distal tubula passes nearby the afferent arteriola of the same nephron, providing the basis for tubulo-glomerular feedback. This figure shows magnification of the contact point between the terminal part of the ascending limb of the loop of Henle and early distal tubula with afferent arteriola of the same nephron. At the contact point, specialized tubular cells, macula densa cells, monitor the composition of tubular fluid, namely sodium chloride, and produce a signal that acts on the smooth muscle cells in the walls of the afferent arteriola. Circulatory system plays a central role in supplying organs and tissues with oxygen and nutrients and removing metabolic waste products. On a large scale, the circulatory system can be viewed as a branching network where larger vessels on the arterial side branch into smaller and smaller vessels until the capillary level is reached. It is clear, however, that there are large differences in the structure of the vascular system among different tissues and organs. This difference relates to specific functions of the tissue and organ. In other words, the vascular topology is optimized to serve the specific needs of a given organ or tissue. In skeletal muscle, the main need is to allow for large variations of total blood flow so that to match the supply with demand during muscular work. In other organs, like the brain, there is no need for large variations of overall flow, but there is a need to redirect flow to local areas where the metabolic activity is high. In the kidney, the critical aspect of circulation is not only to supply tissues with oxygen and nutrients, but rather to support the filtration of plasma at the glomerulus of each nephron. Experimental results show that neighboring nephrons are often arranged in pairs or triplets that creates a basis for their interaction via vascular propagated electrochemical coupling and hemodynamic coupling. Let us consider how autoregulation is expressed at the level of a single nephron. This figure shows a typical 3-day blood pressure recording from a Namatensive rat. Namatensive rat means a rat with normal blood pressure. There are a few major periodicities in the blood pressure. One at the frequency of the heart rate and the other with a period of 24 hours. The mean arterial pressure fluctuates significantly over time with high values at night for a rat and at feeding times. Between these periodicities, the blood pressure displays a 1-to-F pattern. The 1-to-F fluctuations are caused by the independent actions of arterioles in all organs of the body and contractions of large muscles, for example. Variations in arterial blood pressure cause variations in glomerular capillary pressure and as a consequence a tendency for the glomerular filtration rate to vary. The kidneys protect their own function against short-term variations in the blood pressure. The process of autoregulation takes place at the level of individual nephron. There are two main mechanisms of autoregulation. The first one is myogenic mechanism. Myogenic response is how small arteries and arterioles react to changes of blood pressure so that to keep the blood flow within the blood vessel constant. If the pressure within a vessel is suddenly increased, the vessel responds by constriction. Decreasing pressure within the vessel causes relaxation and vasodilation. This mechanism causes oscillations of vessel radius and blood flow with a period of about 5-10 seconds. Cellular mechanism of myogenic response involves depolarization of cell membrane and influx of calcium through voltage-gated calcium channels in response to increasing blood pressure. The rise in calcium activates myosin light chain phosphorylation that causes constriction. The second mechanism is tubulo-glomerular feedback. Tubulo-glomerular feedback is a regulatory mechanism that is specific to the kidney. This mechanism leads to vasoconstriction of the afferent arteriola in response to an increased concentration of sodium chloride at the macula densa in the earlier distal tubula. This mechanism causes oscillations of tubular pressure with a period of about 30-40 seconds. Since both mechanisms act on the afferent arteriola to control its hemodynamic resistance, the activation of one of the mechanisms modifies the response of the other. In this way, slow tubulo-glomerular feedback oscillations provide modulation of faster myogenic oscillations of arteriolar radius. Microperfusion with artificial tubular fluid shows that the pressure oscillations are generated by feedback mechanisms associated with the individual nephron. The first figure shows a tracing from an experiment where spontaneous oscillations in proximal tubular pressure were present. Microperfusion at a rate of 7.5 nanoliter per minute increased amplitude of oscillations without affecting frequency. On stopping microperfusion, amplitude decreased again. The second figure shows a tracing from an experiment where spontaneous oscillations in proximal tubular pressure were present at the beginning of the experiment. Suction with a rate of -10 nanoliter per minute abolished oscillations. The oscillations reappeared again when suction stopped. What else can we record in experiments? Cartoon to the left shows an experimental setup that allows for the recordings of pressure in the proximal tubula and chloride concentration in the early distal tubula. The figure to the right shows a typical recording of distal chloride activity, top trace, and the proximal pressure, bottom trace. Measurements of the early distal chloride activity reveal oscillations with the same frequency as the proximal pressure oscillations. The chloride oscillations show a considerable phase shift compared to the proximal pressure oscillations. The chloride oscillations follow the proximal oscillations with a phase shift corresponding to a delay of 8-10 seconds. A maximum in chloride activity, therefore, immediately precedes a minimum in the tubular pressure. This is in accordance with the oscillations being mediated by tubular-glomerular feedback, because the higher distal chloride activity should cause a tubular-glomerular feedback mediated constriction of the afferent arteriola, and therefore a minimum in tubular pressure. Because the length of half period is about 15 seconds, a delay of 10 seconds between proximal flow and the distal chloride oscillations implies that the delay in propagating the signal across the macula densa and causing a constriction of the afferent arteriola should be about 5 seconds. Additionally, it is possible to measure afferent arteriolar blood flow using laser Doppler probe. Afferent arteriolar blood flow and tubular pressure were measured simultaneously to test the hypothesis that oscillations in tubular pressure were the result of tubular-glomerular feedback-induced oscillations in afferent arteriolar blood flow. The results of a typical recording are shown in the figure. One can see that oscillations present in tubular pressure measurements, top trace, and laser Doppler recordings, bottom trace, are synchronized. The average frequency of the oscillations is about 30 seconds. The average phase shift is 1 second, with blood flow leading tubular pressure. Now the question is whether there are any differences in dynamic behavior of the tubular pressure in non-attensive rats, that are rats with normal blood pressure, and genetically hypertensive rats, that are rats with high blood pressure. These figures show typical recordings. While non-attensive rats show regular oscillations with a period of about 30-50 seconds, hypertensive rats show highly irregular oscillations. Let us discuss now how nephrons interact. It is possible to make simultaneous recordings of tubular pressure in neighboring nephrons. There are two types of interaction. Neighboring nephrons can interact by vascular propagation of electrical signals initiated by tubular-glomerular feedback of each nephron. These signals travel via endothelial cells in the arterial wall and can reach the neighboring afferent arteriola. This coupling tends to produce simultaneous constriction of the afferent arteriolas, leading to in-phase synchronization of the oscillations. Another type of interaction, hemodynamic coupling, occurs due to the topology of renal vasculature. Sharing of blood flow at each branch point of the arterial tree provides an interdependence of the local perfusion pressures, and therefore affects the hemodynamics of all nephrons located downstream of the branch point. Due to a significant time lag in the system, this type of interaction can lead to antiphase synchronization between nephrons. Depending on the precise topology of the vascular network, the first of these mechanisms, electrical interaction, can be more important for the local coupling of nephrons, while the second mechanism, hemodynamic interaction, is more important for global coupling. As we know, nephrons exhibit both tubular-glomerular feedback oscillations and myogenic oscillations. For neighboring nephrons, one would expect to observe frequency entrainment for both types of oscillations. Using frequency condition for synchronization, we can calculate locking time of oscillations for non-attensive and hypertensive rats. This figure shows the probability of finding different time intervals of synchronous dynamics. In non-attensive rats, blue color, the slow tubular-glomerular feedback oscillations have a probability of 40% of remaining synchronized longer than 250 seconds, that is duration that represents 8 or more periods of oscillations. For hypertensive threads, red color, that probability is only 12%. Thus, slow oscillations in normotensive threads are 3 to 4 times as likely to display long locking times as in hypertensive threads. Fast myogenic oscillations similarly have a probability of about 30% of remaining synchronized for more than 50 seconds, corresponding to 8 periods of myogenic oscillations, while for hypertensive threads, this probability is only 12%. One can see that synchronous patterns of autoregulatory dynamics are different in normotensive and hypertensive threads. Thank you.